What do you get when you mix eggs, milk, Gatorade, and nutritional yeast? Well, the answer is, of course, a hamburger. And no, I haven't gone crazier than normal, or at least that's what the voices tell me.
No, he's got a point.
Instead, today we're going to be showing how we can really use these ingredients to grow some living meat. And while we'll only be growing a small amount of cells as a proof of concept today, this work really will set the stage for growing a McNugget in a tank. So, let's get started. This isn't the first time we've talked about growing meat on this channel. Longtime viewers will have seen the meat leaf and meat berry we made, or may have seen the work growing living rat neurons to play the classic video game Doom. The thing that these projects have in common, other than being the eldritch monstrosities that haunt your nightmares, is that they are both examples of modern mammalian tissue culture. Using a special set of techniques, and with the correct solutions, we can not only keep animal cells alive outside of the original animal, but we can use them for our own purposes and grow and multiply them as we wish. But we didn't invent this method. This is the same technique used by scientists around the world to develop medicines and new treatments for patients, or just study how different animals' biology works. The meat leaf, for example, was based on techniques originally invented to try and grow replacement organs for patients in need, though by the same token, a very slight modification of the technique is being used in the manufacture of lab-grown meat meant for human consumption. The point being that the same technique can be used for an almost endless array of projects. Every time we've grown cells like this on the channel, be it the meat leaf or neurons or meat laser or whatever else, you've seen us reach for a bottle that looks like this. These are the standard potions that we buy to keep the cells alive while we work with them. The most common of which is called DMEM, also known as Dulbico's Modified Eagle Medium, which, by now, longtime viewers will know, does not, in fact, contain any eagles. DMEM is actually a mixture of vitamins, electrolytes, sugar, salt, and a host of other nutrients that the cells need.
It's a single-celled protein combined with synthetic aminos, vitamins, and minerals. Everything the body needs.
It doesn't have everything the body needs.
However, DMEM isn't actually complete on its own. Every time we crack a fresh bottle of this stuff, we also add some extra things to it. The most important of which is called FBS, or fetal bovine serum. And yeah, it's exactly what it sounds like. It's baby cow juice, or blood serum extracted from fetal cows. And yes, I hate using this stuff as much as I love saying baby cow juice. Not only is this stuff gross, it's also just really expensive, and carries the risk of contamination with viruses and other junk that inherently makes cell culture less safe. And if lab-grown meat is ever going to take off, you can't be using cow blood to feed the growing meat vats if you want it to be either cost-effective or sustainable.
Feed me. Does it have to be human? Feed me. Does it have to be mine? Feed me. Where am I supposed to get it? Feed me, Seymour.
As such, lab-grown meat companies have spent millions of dollars trying to bring the cost of growth media down, and many of them failed in that pursuit, going bankrupt after years of work. A big part of the reason these sorts of tissue culture videos are so infrequent on the channel is because of their enormous cost. One DMEM and FBS kit to make 500 milliliters costs between $150 and $200, more when you consider the dry ice shipping fee, and it's the cheapest media you can buy. The neuron stuff costs about $600 for a set. And much like your local drug dealer, those prices won't come down because people have to keep coming back for more. And with the nearly infinite budget of a university and grant money burning a hole in their pocket, $600 for a bottle of media and thousands of dollars for a tiny tube of cells makes sense. Since we are not a university and rely on merch sales and ad revenue, and especially the support of our wonderful patrons and channel members, yeah, we don't have that kind of money to burn. So if we ever want to try and make lab-grown McNuggets, or just dramatically speed up our work on the meat robot and other projects, we too needed a better solution. This video, like most of our projects, started with an intrusive thought we let win. When you look at a bottle of DMEM and hear its ingredients, do you not just think, man, that sounds a lot like Gatorade? Because I know for a fact that every biologist I know jokes that this stuff just is fancy Gatorade, and there of course have been the odd Redditor crazy enough to taste it, though I would very much not recommend that. This of course leads to the obvious question. How much DMEM can you replace with Gatorade before it stops working? After all, it's got what cells crave.
Brondo's got what plants crave. Yeah, it's got electrolytes.
What are electrolytes? Do you even know?
And if Gatorade can replace DMEM, what common grocery store ingredients might be able to replace FBS? Well, today we're going to find out. Whatever we learn from this will be used to design our own complete replacement that we'll use in the next video. And next time, we can just focus on growing a huge amount of cells. Our bioreactors are just itching to fill with that sweet, sweet McNuggety goodness. Thankfully, we're not the only ones who let the intrusive thoughts occasionally win, because a group of Japanese students actually ran a version of this experiment several years ago, which gave us a great jumping off point. They tested a few drinks beyond just Gatorade, including 2 Japanese ones called Green Dakara and Bakari Sweat, and they found, to their amazement, that they could replace, in some cases, up to 70% of the medium and still have the cells grow. As such, we popped to the store and bought a variety of drinks, and I also imported the 2 Japanese ones from the earlier work. Now, there was a little wrinkle with this, because while the drinks themselves were about $2 or $3 a bottle, shipping what amounts to several pounds of water from Japan to Canada costs several $100s. So in future, I'd recommend looking for a local supplier if you want to replicate our experiments. The drinks we settled on are Cool Blue Gatorade, Melon Pineapple Powerade, Kiwi Strawberry Vitamin Water, and the 2 Japanese ones, which only come in 1 flavor each. The plan is to make a dilution series, where each well of a 12-well plate has an increasing amount of the drinks versus DMEM concentration, to see at what point the cells first stop growing properly, and then at what point they just start dying en masse. It was both hilarious and delightful having to handle bottles of Gatorade in a sterile flow hood. One of the fun things is that because these drinks are so full of sugar, they definitionally must be sterile, otherwise they would just rot on the shelf. So, as long as the bottles are sprayed down with alcohol before bringing them into the hood, we could just use them directly without having to further sterilize them. As I started making the plates, the laughs just kept coming as a rainbow of wells started to form as the dyes and pH indicators both in the DMEM and drinks interacted. And when everything was said and done, we were left with 5 plates that just needed cells. But before we do that, we noticed that a lot of the drinks were making the pH drop a lot, which would be bad for the cells. So, to remove that as a variable, we made a whole second set of plates, but this time we took time to tediously pH balance a sample of each drink to make sure they were neutral before use. Suffice it to say, it was enough slow, careful pipetting that I needed some coffee just to stay focused. Coffee I drank out of my wonderful Pipette Cry Repeat mug, which is only one of many amazing items we actually carry in our new store. If you want to help support the channel while showing off your mad science flair, head over to thethought-emporium.ca and snag something after this video. And for those that missed the initial sale, our handmade papyrus mummy assembly instructions are now back in stock. As a little bonus, if you can find the hidden code somewhere in this video, you'll get 10% off your whole order. Okay, cell time. Today we're going to be using mouse dermal fibroblasts as our cell of choice. You've seen us use these ones before, and the reason is simple. Fibroblasts are fairly robust cells and also grow very quickly, so we'll get our results just as quickly. Also, their morphology can tell you a lot about their health, so it should be really obvious if they're healthy or not. And also, they're literally the cheapest cells you can buy. But for those worried about how this will apply to other cell types, thanks to the work of the lab-grown meat crowd and others, you find that in reality, most cell types can be grown in basically the same media. The only reason academics use the proprietary mixtures sold by most biosupply companies, as far as I can tell, is basically just tradition? Because when you actually test it, DMEM and FBS works for almost anything. Save the occasional addition of 1 or 2 bonus ingredients if you're growing something really weird like bones. But that's mainly just because they need really high levels of calcium, as you might imagine. And that makes sense, right? Since it's not like you have 100 kinds of blood, you just have blood. Anyway, to get things started, I set up a fresh flask of fibroblasts as I've shown how to do many times before on the channel. These were allowed to grow for a few days in the incubator in advance of our Gatorade experiment. Once all the Gatorade plates were ready and warming in the incubator, I used a solution of an enzyme called trypsin to make the cells release from the flask so they could be collected. Over a few minutes, the cells go from flat to floating round balls once they let go of the plastic. After a few minutes of enzymatic treatment, they were all floating around, so I transferred the suspension to a sterile tube to be spun down and collected. The trypsin solution was then removed and replaced with pure fetal bovine serum. This is a bit odd, as normally we'd resuspend in culture media, but this way we can introduce the correct amount of FBS to each well, and an identical amount of cell material without diluting anything. We set the ratio so that we'll be adding about 100,000 cells in 200 microliters of FBS we need to add to each well. Alright, while those grow in the incubator, we can turn our attention to the fetal bovine serum replacements. Because while we were dreaming of the similarity of Gatorade and DMEM, we started to think about what other common items could replace fetal bovine serum. I mean, what is serum really? Serum is just blood without cells, so what else is in the liquid? There's nutrients, of course, but the DMEM kinda takes care of that side of things. There's some hormones, and also lots of albumin, which give the cells something to stick to. And finally, there are some specific growth factors. These are special proteins that bind to the cell and tell them to divide. Now, to be clear, I don't actually have an issue using animal products. Fetal bovine serum is just cruel and inefficient. But what about something like milk? A cow is a machine that turns grass into milk and can live a very comfortable life doing so. Or what about chicken eggs? It's very easy to raise chickens in a humane way, and they'll produce eggs every day for years. If these things could be made into FBS replacements, they could produce more meat than could ever be generated from that one animal's life. One chicken could make hundreds or thousands of chickens worth of meat, and it doesn't even need to die to do so. And when you think about it, milk is basically just purified blood with fat, protein, and bonus sugar mixed in. So skim milk, or possibly whey, should contain a lot of the same growth factors as blood. And yes, think about that a lot when you're having cereal tomorrow. Eggs, on the other hand, literally contain everything a growing chicken needs, so it almost certainly contains the right stuff, and a quick perusal of the literature actually confirms high levels of all the same growth factors that are used in lab-grown meat production. But to be sure that we don't miss something at the grocery store that might work for reasons that we hadn't considered, we also grabbed a smattering of other odd liquids to try. These include coconut water, beer, and Maggi sauce. We also picked up chicken broth, beef broth, and bone broth to see if rendered meat still contains the right stuff, though obviously we wouldn't want to use that long term. And as a comparison, we also included the synthetic chicken and beef soup that we made in a previous video. Now, almost none of these liquids could be used straight and needed some processing. At the least, everything needed to be loaded into a centrifuge tube and spun down to remove any particulates or chunks of stuff. And then they each need to be filter sterilized. This means loading the purified liquids into syringes and then squeezing them through a filter that has holes that are so small that no living organism can fit through. But some of the liquids need more work, the eggs especially. To get started, we needed 2 eggs. The first was carefully separated into the white and yolk to be processed separately, and the second is just opened into a beaker. We started by transferring the egg white to a blender and homogenizing for a few minutes. When it's nearly at the stiff peak phase, we added 5 grams of regular white table sugar and blended for another minute. This mixture was then transferred to a piping bag and distributed on a baking tray, before baking for a few minutes on high. Once cooled, you'll be left with a lovely meringue. Wait, what were we doing? Oh, shit, right, cell culture, my bad. I meant to say that we dilute each egg fraction with 5 times the amount of water and then blend it for 30 seconds to fully homogenize the mixtures. These were then transferred to centrifuge tubes as before and spun down on max speed to remove any solids. And, like the others, they were filter sterilized, though the thickness of the liquids made that rather tedious. Finally, for the milk, we also prepared fractions of whey by acidifying some skim milk with vinegar to make all the fats and proteins crash out. This too was centrifuged to remove any remaining solids and then pH balanced back to neutral before filter sterilizing. To actually set up the experiment, we prepared 3 plates with normal DMEM, plus either a 50-50 mixture of fetal bovine serum and one of the test liquids, or just the pure test liquids. In both cases to a concentration of 10% of the culture volume. And of course, once that was done, we added an even amount of cells to each well. These plates too were added to the growing stack in the meatcubator and were left for 48 hours to grow. This way we'll know it's not a fluke if the cells are still alive after 2 days. So, how'd everything do? Well, I'm not going to have you sit through an inspection of every single well, but I will show you some highlights. Also, to make it easier to gauge cell health, we used a special stain called calcine AM, which is what's called a live cell stain. In its base form, it doesn't glow or respond to UV light, but when it diffuses into a cell, enzymes will modify it, converting it to a fluorescent form. So the inside of the cell will get stained bright green, and dead cells should stay colorless, as any dye that does get in will quickly leak out through holes in the dead cells. Or, if they're only mostly dead, they'll look like little floating round green balls.
Look who knows so much, huh? Well, it just so happens that your friend here is only mostly dead. There's a big difference between mostly dead and all dead.
Okay, so here's a control well of cells. You can see that the cells are all nice and flat and spread out and are, of course, bright green. There's also very little debris floating around or the little balls of dead cells. Normally, after 24 hours, we'd give the cells a rinse to remove any of these initial floaters, because always there's a few, but we didn't do that as to not disturb the cell count so we can see what actually survived. Now, how did the drinks do? Well, starting with the regular, non-pH balanced ones, Gatorade and Powerade did surprisingly well. Here's Gatorade at 40% concentration. The cells look pretty healthy, nice and green, good morphology, not too much dead stuff, but at 50% there was a sharp fall off and everything dies. Powerade was similar, good until 50% and then death. Vitamin water was a standout, as even at 10% concentration was almost completely lethal to the cells. So whatever is in there, they do not like growing in, but that could just be one of the flavorants. Though do keep this in mind the next time you see an article about how something kills cancer cells in a petri dish. Lots of things kill cells in a petri dish, but you could drink a gallon of this stuff and mostly just have a stomachache. Now for the real surprises. The Pocari Sweat didn't really do any better than the Gatorade or Powerade. Here's at 40%, they look good, and at 50%, not so much. But the Green Dakara was way better. The cells looked great at 70%, but start dying by 80%. The repeat experiment with pH-balanced drinks showed a similar trend. Now, the cells could survive everything, other than vitamin water, up to 60 or 70%. But the Green Dakara? Here are cells grown in 90-10 Green Dakara to DMEM. They look nearly perfect. Sure, some debris, but that would get washed out. This was a frankly stunning result, and quickly set off a chain of digging through what the hell is actually in DMEM and especially Green Dakara to figure out why this worked. If you happen to have a store near you that carries this stuff, your DMEM cost just dropped by a minimum of 70%, up to 90% if you want to live dangerously. But we won't be stopping there. We're going to conquer that last 10% and make a 100% replacement, but now let's look at the FBS results. Here's a graph of the results, and we'll look at some images in a second, but the way we did this was on a 5-level rating scale. A 5 meant the cells were indistinguishable from the control. 3 is at least half the cells are dead, and 1 meant that a few cells survived, and 0 is total destruction. Right away, we see a few standouts and a bunch of surprises. To our shock, at 50%, beer, while the cells were not happy about it, they did grow. And it did about as well as bone broth, skim milk, and nutritional yeast. On the other hand, all the egg fractions did great, as did coconut water, and what really surprised us was the fake chicken soup from our previous video. And it performed better than actual chicken soup, which is also how it performed in the taste test. Maybe the cells just wanted more umami? The 100% tests were a little bit more grim, but we did still have some success. The best by far were the whole egg, pH-balanced whey, and nutritional yeast. Each looked a little rough, but the cells were alive. So while this isn't a home run, it does point the way to how we could achieve much better results. Because of how well this and the Gatorade test works, it seemed like the gap between cheap and easy media and what we already have is pretty slim. Most likely, we're just missing a couple of ingredients, or the level of something isn't right. Alternatively, there's some cell inhibitory compound in some of these that's skewing the results. Remember, something in vitamin water was super lethal, yet its ingredient list is nearly identical to Powerade or Gatorade. Perhaps we just need to process the eggs or milk differently, or we maybe need to add a missing growth factor or two. Maybe it's as simple as just adding insulin, which is one of the only things that we couldn't get from the store-bought ingredients of the things that are normally found in FBS. Another option we didn't explore here, but may in the future, is just straight up growing all the growth factors we need. This is the approach the lab-grown meat crowd are going with, and there's a fair bit of literature looking into what's actually in FBS and which growth factors you really need. Industrially, the plan is to just grow those special proteins transgenically in something like E. coli and then extract them. It's a lot of work, but it's not a difficult or particularly expensive process, and our lab is already fully kitted out to do exactly that. So for us, the only thing we would actually need to try it are the 3 or 4 pieces of DNA off Ad gene, or to just get the DNA printed ourselves. Next time, whatever the case, we'll have the recipe dialed in and can focus on bulk cell growth and really growing an edible piece of meat. The only reason we haven't already attempted that was the insane cost of store-bought media, though that'll also mean needing new sources of cells. Speaking of which, if you've ever wondered how we go from Timmy the Rat to Timmy the Tube of Cells, we're working on a video demonstrating the process. But even though it's a standard laboratory procedure, YouTube's content guidelines would make it very risky to post. This is a recurring problem in our work, where things that every bio student does in their first couple of years of university labs would just be far too gory for YouTube. There are techniques and methods that are vital to modern biology I just don't feel like I can show here. Even carefully researched and documented projects filled to the brim with art and science can still get demonetized for no reason. For example, our recent video where we spent 9 months perfectly recreating the Egyptian mummification process got demonetized even though we were using a store-bought chicken from the grocery store. And that's one of the many reasons that I am so glad to be part of Nebula. Nebula is the amazing creator-owned streaming platform where all of your favorite creators have come together to make and share the content you love, without having to worry about advertisers. Not only are all of the videos completely ad-free, there is a ton of bonus content there too. Our mummification video, for example, came with a bonus 25-minute deep dive into the recipes, spells, and hieroglyphs used in the project. Beyond those Nebula exclusives, there are also Nebula originals, like Real Science's Becoming Human series, where they explore the origins of the first technological leaps of our species. And if that wasn't enough, there are also Nebula classes, where creators teach you about a wide range of topics. I've particularly been enjoying Devin Stone's Copyright for Fun and Profit, which really helped clear up what copyright law can and can't do. Nebula is the fastest growing independent video platform, and we are joined by so many amazing creators, like Nile Red, Breaking Taps, Strange Parts, and too many others to possibly list them all here. Nebula is also helping creators like us elevate our content and take it to the next level. Better production value, bigger stories, all without having to chase the trending tab or appease the whims of advertisers. So if that sounds good, then use the link in the description to get a year's subscription for the incredibly low price of $30, or just $250 per month, or the best deal of them all, which is a lifetime subscription for only $300. I literally can't think of any other streaming platform that is as good value for the price. So head over to nebula.tv/thethoughtemporium to sign up today and upgrade your viewing experience. That's all for now, and we'll see you next time.